Let's learn about the immune response. The immune response is the response of our white blood cells to an invading pathogen. A pathogen can be a microorganism with antigens on its surface, and by definition, a pathogen is a microorganism that causes a disease. Okay, so we're going to start off with our pathogen over here, outside of the body, with its very particularly specifically shaped antigens on its surface. Antigens, as you've already told me, are protein molecules. They have their primary, secondary, tertiary structure. Okay, very specific shape. This is our skin. Now, if the pathogen enters our bodies, then our immune response kicks in. Okay, so let's take our pathogen, let's let it enter our body. Okay, now just so we're aware, these pathogens, they can multiply quickly. Okay, so the number of pathogens when they invade our body is going to increase quickly. So we're going to need several layers of kind of attack or defense to kind of combat this. Okay, the first kind of response is from our phagocytes. Okay, this is our phagocyte. It's a type of white blood cell. We came across phagocyte at GCSE. The phagocyte has inside it a vesicle that has hydrolytic enzymes. Or lysozymes. Okay, and the pathogen, when it enters, well, it's releasing chemicals. It's just giving out some chemicals, right? Now, the chemicals are at a high concentration near the pathogen, but they become present in a low concentration the further away you go. Okay, so the concentration gradient of the chemical secreted is high near the pathogen, but low the further away you get from the pathogen. And what the phagocyte does, it just detects that very, very, very low concentration of that chemical that's being released. It just detects that tiny low concentration. And that's enough. Because then the phagocyte moves against that concentration gradient. So let's take our phagocyte. It moves against that concentration gradient towards the pathogen that it has recognized as non-self. The antigens are recognized as non-self, don't belong to the body. Upon reaching the pathogen, what the phagocyte now does is it changes its shape. It extends around the pathogen, ultimately to engulf it. Remember that word, engulfs the pathogen. The white blood phagocyte goes and surrounds, and it forms this structure here called a phagosome. So the phagosome is that bit in the middle where the pathogens are. Okay? What happens next is this vesicle here with the hydrolytic enzymes will fuse with the membrane of the phagosome, as so, and the hydrolytic or lysozyme enzymes are released onto the pathogen and break it down. Break it down. The pathogen's gone. But the antigens are still there. Okay? What the phagocyte then does is let's do another thing here. Let's add one more step. So when the pathogen is broken down, it's broken down into soluble molecules or parts and insoluble parts. Okay? Now the soluble part, ultimately, this is molecules, isn't it? Or parts of molecules. Some of those, the phagocyte might be able to use. Or if they're soluble, maybe gets rid of it in that way. But the insoluble parts of that phagocyte are removed from the cell by exocytosis. Okay? Now, let's go back to this antigen. When the phagocyte is broken down, antigens are taken and presented on the surface of the phagocyte. Okay, so the phagocyte has on it molecules where it can present, like these are like receptor molecules kind of thing, yeah? Okay, so you remember the membrane, phospholipid membrane, got proteins that can have like lipid or carbohydrate chain on it, acting as a binding site or cellular communication or as a receptor or whatever. Okay, so the phagocyte has these on its surface, and the pathogen, the antigens for a pathogen are presented, okay, on the surface of this phagocyte, making this phagocyte now an antigen presenting cell, APC. Okay, now this is very important because we need the very specific antigens of this pathogen in the system because we want to destroy that particular pathogen, and that's the only way we're going to recognize it. The only way we're going to know that that pathogen is there to be destroyed is by identifying that that has those specific antigens on it. So then our immune system now needs to do something to ensure that we can now find the right pathogens to now kind of destroy them. So what happens over here? We have a collection in our bodies of what we call T helper cells. And we have different types of T helper cell. Okay, we have T helper cells with a variety of different molecules on its surface. And it's the T helper cell with the complementary molecules on its surface that will attach to the APC. So this one, it's not complementary. It's not going to attach. This one is, okay, should have drawn a bit bigger, but you get what I'm saying. Is complementary. So it is going to attach. And that's a great idea. That's a great idea. Because now what this T helper cell does, because it has receptors on it that fit or bind with that particular antigen, that T helper cell now can do one of four things. Right? One of four things. First and foremost, well, they're all alongside each other. They're all equally important. That T helper cell goes, okay, I'm going to become a memory cell. I'm just going to remember that that was the shape of the antigen for that particular pathogen, so that in the future, if we need to make antibodies to kill this pathogen, this design of antigen, we're ready. So that one option, the T helper cell becomes a memory cell, so we can remember later. And remember, what we have is our immune response. And we have here maybe the number of antibodies, and we have the first exposure to a pathogen where we make the antibodies gradually and slowly, but we get there in the end, right? So we might have symptoms for a bit longer. But if we are exposed again to the same antigen, because of our memory cells, we can then defend against them a lot more quickly. So we can actually make more antibodies in less time because the T helper cell changed into a memory cell. Okay, so that's step one. There's something else that the T helper cell can do. It can stimulate beta cells or B lymphocytes to divide. And we'll come back to that one in a minute. Okay, because it's these guys, the beta cells, that are going to go on and make antibodies. Okay. This T helper cell can stimulate more phagocytosis. Sorry, stimulate more. Phagocytosis, which can only be a good thing, because then we've got more phagocytosis, more APCs, more T helper cells, so this is going to happen more. And it can activate cytotoxic T cells. Now, these cytotoxic T cells release, so these are cytotoxic, and these cells release an enzyme called perforin, which acts to destroy. It actually makes holes, perforates the cell membrane of the pathogen, destroying it. So it's inside spiller. Quite a ruthless kind of enzyme. Okay. So that is the work of the 4 different things that specific T helper cell will go on and do. Okay. Now, if we have a lot of B cells, we can make a lot of antibodies. But we're going to make specific antibodies so that we can destroy that particular pathogen. They're going to be antibodies that are a particular shape. Okay. All right. So let's go with how these B cells make antibodies. So I'm just going to take this bit here, the B cells, and how they make antibodies, and we're going to just put it down here. Okay. So if there are lots and lots of B cells, that can only be a good thing. Because what the B cell will do, so we have our B cell here, and there's lots of them. It takes the antigen, okay, from the pathogen. So we have the pathogen that's invading, okay, and the B cell processes that pathogen. Don't worry about that too much. It interacts with that pathogen and takes the pathogen and puts it onto its surface. Thereby, it becomes also an APC, presenting the antigen on its surface. Okay, so let's just take this here. Let's put another one here. Okay, and remember that T helper cell with a specific shape? It comes along and it goes, okay, B cell, I see that you've got the antigen on your surface. I'm now going to bind with the antigen, and by binding with the antigen, I'm going to activate you to make lots of yourself. Right? So the T helper cell activates this B cell to make lots of copies of itself by clonal, or just, no, by cloning. Sorry, not clonal selection, cloning. Gets it to make lots of copies of itself, cloning by mitosis. Okay. And then these B cells can do 2 things. They can become plasma cells, right, which will make the antibody that we needed. Remember, matching the shape, and thereby releasing these antibodies so that they can then go and destroy the pathogen. Or they go, okay, I'll become a memory cell too. So I remember what I did next time. Okay. Let's look at the work of the antibodies now. and how the antibodies interact with the pathogen. So the whole point is to make lots of antibodies, right? How do the antibodies actually interact with the pathogen? That's what we're going to look at next. Okay, I'm going to kind of take this over here. All right. To understand this, we need to appreciate that the antibody has a very beautiful shape. It's very specific. Antibodies, protein molecules, again. And they have this kind of like structure where they have long chains and they have two short chains. These are the constant regions. These always stay the same. Okay, there's a bit of a disulfide bridge going across the two long chains here. But it does have, at the end of each of the long and short chains, a variable region. Okay, a variable region. And it's that variable region that is specific to the shape of the antigens. Okay, and what that antibody does is it can bind with the surface of the antigens on the pathogen. And it will bring the pathogens into the same space. It will bring the pathogens into the same space. So one variable region get that pathogen, one will get that one, and bring, rather than all swimming around everywhere, let's just get them in one place. Because if we can get them in one place, then we can break them down more effectively, more efficiently. And that's the job of the antibody. The process by which the antibody binds and brings the pathogens to the same space or near to each other is called agglutination. Okay? And that, ladies and gentlemen, brings me to the end of the story of the immune response.